I tell you, so we not no time for no small talk. We just jumping right back into it like if we never done. But actually, we have a little time for a little small talk. Like we run the sponsors here. Boom. Sign up for CSEC and keep crash courses. WhatsApp this number to the student. But most of our crash courses done. We have physics. Most of our crash courses done. We have some keep crash courses. You still have keep what? Unit 2 or Unit 1, Akili?
Unit 2, Unit 1 went
Unit
already.
2. We have a couple crash courses left over. So y'all could jump in on those. But what you can do is make note of this number. If you are watching this in any time past July, you could sign up for the Student Hub online classes. Right now, we're doing a revision session right before exam, and some people on the zero. And we're trying to bring them to hero, zero to hero. So if you sign up to the Student Hub online classes in July, August, you wouldn't have to come from zero to hero. But nevertheless, we helping anybody we could. Well, now we Akili helping anybody she could on biology, and we just doing a kind of crash course through biology here to help you for the exam in the morning. Remember to take notes, but if don't let that not help that stop you, sorry, from focusing, because we already send the notes on the Discord. If you're on the Student Hub Discord, it's public. You can access it on Instagram, link in the bio. The notes is on the biology channel, so you can just jump into that and you can follow along with Miss. All right, that is it for me. I'll enjoy.
All right, so we're back. We were in the middle of the circulatory system. I am going to preface by saying this, like, it's a lot of content I gave you guys in the notes. I do not have the time to go through all of that in detail tonight, right? So we're going to go through as much of it. I hope that having the notes means that we could move at a little faster pace. I left all of my annotations up, so that means I have less writing to do. Those of you that are talking like you're cooked in the chat, remember your mindset is going to be a very important part of it. The more downcast you are, the slower you're going to be able to absorb the information. So come at it from a different mindset, a better mindset, more of, just equipping yourself to win. And I am going to help you do that as the mandate of the Student Hub, right? So let's continue. The only chambers, I told you that in when the heart is beating, your both atria are going to contract together, your both ventricles are going to contract together, and the pulse that you feel around your body is in sync with the contractions of your ventricles. One thing to note is that the walls of your left ventricle is going to be thicker than the walls of your right ventricle, and that is because of where where those ventricles are pumping blood to. Your right ventricle is sending blood to the lungs, which are nearby, so it doesn't need to build up that much pressure or do that much work. Whereas your left ventricle is pumping blood into the aorta to send blood all around the body. So therefore, it is doing more work than the right ventricle. So the walls of your left ventricle is going to be thicker than your right ventricle. The next thing I want to highlight is that you have various valves within the heart. You should know those valves. So we already identified the tricuspid valve, the bicuspid or mitral valve. You're going to have these two are semilunar valves. You have your aortic valve and your pulmonary valve. And valves have the same job no matter where you're looking at it. It keeps blood flowing in one direction, right? So valves in the heart, valves in your veins, they're all about keeping blood flowing in one direction, right? So looking at that, we move on to this figure, which is showing you blood vessels used to transport blood around the body of a mammal. And this here, you have blood vessel A and B. They're asking you which one represents a vein. Which one do you think is going to be representing the vein? Right? So because B has the valve, that is going to be the one that is the vein. Your veins transport blood at much lower pressures, which means that you're more likely to run into the occurrence of blood wanting to flow in the reverse direction. So as a result, your veins are going to have valves in order to keep blood flowing in one direction towards the heart. Your arteries don't need valves because they're transporting blood at high pressures. And you need to note some of the structural differences between your artery and your vein, right? So we know that this one is a vein. One structure in the vein is going to be that valve, right? This is your artery. This is your vein. Note that I changed the color of the background of the slide so that we wouldn't have any visual issues. right? Your arteries, you look at the walls. The walls of your artery, the walls of your arteries are much thicker than the walls of your veins. And again, that has to do with the pressure of the blood that it's transporting, right? So your artery is going to be taking blood away from the heart. It's going to split into arterioles, which are smaller arteries that then split into your capillary networks, right? So these will be your capillaries. Your capillaries have adaptations. They're only one cell thick. They are highly branched. They're going to bring the blood as close to all of your respiring cells as possible. And then those join back up into venules, which are smaller veins that will join back up into your larger veins, right? So just a note that about the structure of the walls. So arteries are away, veins are two in terms of the heart, right? Looking at blood, so the last component of your circulatory, you have your heart, you have your blood vessels, and then you're going to have blood, right? And the components of blood, you need to be able to identify the various components of blood. So it's going to be what is A pointing to, what is B pointing to, what is C pointing to. The PDF was posted on the CSEC Bio page on the Student Hub Discord, right? So A is going to be pointing to your platelets, B is going to be pointing to your white blood cell, and C is going to be pointing to your red blood cell. Anytime they give you like a smear like this or a picture of the blood, the most abundant cell type is going to be a red blood cell, right? That's the most abundant cell type. Then less frequently you would have your white blood cells, and then the smaller specks that you will see, which are fragments of cells, would be a platelet. So that's how you identify it. Name the component of blood that is responsible for clotting that you're seeing there. It's going to be your platelets. Your platelets trigger your clotting process, right? So they said, name 3 components of human blood that's going to be important for protecting the body against pathogenic diseases and state the functions of each. So one, we can lead with the platelets that we just discussed because this is a different part of a question, right? So your platelets aid in the clotting process. They also form platelet plugs, which are in case the break in the skin, etc., is small, is going to be able to stop bleeding. But even in larger cases and larger breaks in the skin, it's going to trigger the clotting process in order to prevent, to minimize the loss of blood and also to block the entry of your pathogens. Pathogens are going to be microorganisms that cause disease. Your white blood cells, they have different approaches because you have different types of white blood cells. But you're going to have your phagocytes, which surround and engulf bacteria. So it surrounds it, brings it into itself, and then digests it down to its basic components. Those are going to be your phagocytes. Also, your lymphocytes are going to produce antibodies that are unique or specific to proteins found on the surface of those pathogens. So that's your white blood cells. And then your antibodies, which are produced by your lymphocytes. Well, these cause your foreign organisms to stick together so that your phagocytes can surround, engulf, and digest them, as well as those white blood cells can produce antitoxins, which can neutralize toxins produced by your pathogens as well. Some people ask for transport in plants as well. So you have figure 6 showing transport vessels of a flowering plant. Identify vessels at positions P and Q. P is going to be what? Q is going to be what? Sanjeev, I don't have diseases in the queue. When I polled for topics, nobody asked about diseases, right? So P is going to be a xylem and Q is going to be a phloem. A big clue is going to be the direction of flow. Flow in your xylem is always upwards, right? Because by a transpiration stream, so movement in your xylem is always going to be upwards, whereas in your phloem it can be upwards or downwards, but never both in the same phloem tube, right? So explain why plants do not need an organ like the heart in their transport system, and outline one mechanism by which plants move fluids through their transport vessels. So we're going to discuss the answer here. So what you have to be able to do, or make sure that you remember how to do, is that to explain or to compare and contrast the blood vessels in your mammals versus your plants, right? So plants have a lower metabolic rate, so they do not need substances as quickly as your animals are going to. That's one reason. Loading of your sieve tube generates a high pressure in the tubes at the source. The source is where you're going to have your photosynthetic products being made, whereas the sink is going to be where those are going to either be stored or they're going to be used, right? So you need to get those products of photosynthesis from where they're being made to where they're going to be used or stored. And the pressure gradient generated within your phloem tube is going to be responsible for movement in that direction, right? Briefly describe transpiration in plants. Your description must include the name of 2 organs involved in transpiration. So the transpiration or the transpiration stream is what explains why water and minerals moves upward in your xylem. You have other processes like cohesion and adhesion, root pressure, all helping, but the main explanation is going to come from your transpiration stream, right? So water evaporates from the surface of cells into the air spaces and diffuses out of the plant out through the stomata of the leaf, right? So that water vapor is being lost. That water that is lost has to be replaced by water from your xylem. So that's going to move water from the top of your xylem, dropping the hydrostatic pressure at the top of the xylem. Now, that means the hydrostatic pressure at the bottom of the xylem is going to be high, and the hydrostatic pressure at the top of the xylem is going to be low. So that pressure differential, that difference in pressure, is going to cause the upward movement of water in the xylem, right? Water then moves up the xylem due to the pressure differential, right? And then water is then going to move into the roots to replace the water lost from the base of the xylem. So now that the hydrostatic pressure drops at the base of the xylem, due to water moving upwards, water is going to move from the soil into the root hairs, from the root hairs into the root itself, until it gets to the xylem to replace that water that's being lost. So that entire connection from soil all the way to the atmosphere, where we have water moving through the plant, that is going to be your transpiration stream. Moving on to excretion. So if we're going to be talking about excretion, we're mainly going to be talking about the nephron. We're going to be looking at the excretion of urea and other compounds that's going to be in urine, right? So figure 7 is a diagrammatic diagram of the nephron found in a human kidney, right? So we have to identify the structures labeled A, B, and C, and then briefly describe the process that is taking place at each of these structures, right? So I'm going to go through the whole, what is happening at each phase, and then once we get the complete overview, you should be able to answer what is happening at A, B, and C, respectively, right? You need to do the nephron in detail, right? So this cup-shaped structure here is going to be called your Bowman's capsule, right? So the blood is going to be brought via an arteriole, and it splits into a ball of capillaries known as the glomerulus that fits into this cup-shaped structure, right? So that means plasma and other substances dissolved in plasma, and once it's small enough, it is going to filter into the Bowman's capsule via a process known as ultrafiltration, which is filtration under high pressure, right? Once it's in the Bowman's capsule, it's going to be called filtrate, right? That filtrate has to flow through the nephron. So the first thing, after it leaves the Bowman's capsule, that filtrate is going to flow through this convoluted region here known as the proximal convoluted tubules. So you have here, so A is going to be a Bowman's capsule. Oops, that was supposed to come up stepwise, but it's all there, right? So A is going to be a, so this is going to be a Bowman's capsule. It's then going to flow into your PCT, which is your proximal convoluted tubule. In your proximal convoluted tubule, the majority of reabsorption happens there. Now, remember, in that filtrate is going to have a lot of things that you need to get rid of, but it's also going to contain a whole bunch of beneficial substances that you really just can't afford to lose. Glucose that you work hard to break down, amino acids that you work hard to break down, some ions, water, all of those things cannot be lost from the body every time blood passes through the kidney. So we have to do a lot of reabsorbing. And the majority of that reabsorption is going to occur in that PCT. 60% of your water needs to be reabsorbed there. All of your glucose should be absorbed there. All of your amino acids should be absorbed there. So your proximal convoluted tubule is the main site for reabsorption. This loop here, or this U-shaped part, is called the loop of Henle, right? And basically what is happening in these two sections here is that the part that is coming down, this is known as your descending loop of Henle. The part that is going up is your ascending loop of Henle. And the longer this is the more concentrated urine that can be produced because it allows for more reabsorption of water, right? So then after you pass through the loop of Henle, and you have different things happening in the descending limb versus the ascending limb. So the descending limb, water leaves. In the ascending limb, you're going to have ions leaving the filtrate. It's then going to flow through your distal convoluted tubule, and then at X, it's going to enter into something known as your collecting duct. Right? Your collecting duct is going to be receiving the filtrate from various nephrons. And in that collecting duct is where your antidiuretic hormone, which is the hormone that helps with osmoregulation or the regulation with the amount of water in the blood. Antidiuretic hormone makes your collecting ducts reabsorb even more water from the filtrate, which means that the urine that's going to be produced would be a lot more concentrated. So your brain is going to release antidiuretic hormone when your blood is too concentrated, when you need to reabsorb more water, right? So you have to know the role of antidiuretic hormone in osmoregulation within the kidney. Moving on to irritability. So this says, with the aid of a diagram, describe how a nerve cell is adapted to carry out its function, right? So we're going to look at this motor neuron here, and you have to be able to draw this diagram and label it, right? So drawing this diagram, so here, this motor neuron is going to be receiving electrical impulses via dendrites, right? So it's going to allow it to be able, having multiple dendrites, it's going to allow it to be able to receive impulses from multiple other nerve cells at the same time. It then brings it to the soma or the cell body, and then it's going to go along the axon. The axon is going to be this long extension from the cell body that is going to allow the electrical impulse to travel over great distances quicker. Then around the axon, you have these Schwann cells. These individual Schwann cells are basically other cells that just grow and loop around the axon. So because it's growing in loops around the axon, it builds up a thick layer of phospholipids around the axon. And what it does, it produces the myelin sheath, which is insulation for the axon. Now, the electrical impulses that's being traveled along the axon are a flow of ions, right? It's a flow of sodium and potassium ions. So by putting the myelin sheath, growing this myelin sheath around the axon, you're going to have, it allows the speed of transmission to increase so that the electrical impulse can travel along the axon that much faster. Once the electrical impulse reaches to the terminal buds of the nerve cell, it's going to stimulate the secretion of your neurotransmitters, which are going to communicate either with another neuron, or it can facilitate communication with, for example, your muscle, right? So this is just a recap of how you have electrical impulses being conducted along your nerve cell and how it's adapted for that process. Then it says, with the aid of a diagram, discuss a reflex arc, right? So this is how your different types of neurons and other components are going to come together to facilitate the communication with your central nervous system and your environment, right? So I didn't sketch a diagram, I just pulled one from online, but your diagram here could simply just be terms, right? So there are different components that you must have in a reflex arc, right? So you must have a receptor, right? So the receptor is going to detect the change in the environment. It's going to detect the stimulus. So in this case, it told us that the stimulus is going to be a hot surface. So stimulus is a change in the environment. The receptor is going to be the component of, you don't have to draw the arm. As I said, you could just use the terms or you can sketch, right? The receptor is going to detect the change, the stimulus, the change in the environment. That is then going to send a message to your CNS via your sensory neurons. Sensory neurons take electrical impulses towards the CNS, right? So once that electrical impulse comes towards the CNS, then the component of the CNS that's going to be involved here is going to be a spinal cord. Your central nervous system is your brain and your spinal cord, and some reflexes involve the brain, some reflexes involve the spinal cord. Talking too quickly. All right, I'm going to slow down, right? So once the CNS makes a decision, it's going to send instructions to effectors. That is going to be taken by your motor neurons. Your motor neurons are going to be taking electrical impulses or messages away from your CNS and towards your effector. So in this case, it's going to take this message to this muscle. It's going to tell the muscle, A, you need to contract, which is going to bend the arm and remove the fingertips away from the hot surface to prevent damage, right? So this is your reflex arc. Now, a lot of you are in the comments saying, well, my teacher didn't teach this, etc. But these are past paper questions. These are things that have been asked in previous exams, which means it's on the syllabus and it's fair game for CXE to ask, right? Somebody asked me to recap the different regions of the brain. So state one function of each of the following regions of the brain. Your cerebrum is responsible for conscious thought, logical thinking, right? So that's it there. Your cerebellum is going to coordinate muscle movement and balance. And your medulla coordinates involuntary action. So that's going to keep track of influencing your heart rate, your blood pressure, your breathing movements, because those are things that you are not going to want to have to keep to the forefront of your mind to keep remembering to do, right? Then that brings us to the eye. You do need to be able to identify or label the various structures of the eye, right? So you have A, B, C, D, E, F, G, right? So here you had to identify the letter. So they give you a list of functions and you have to see which letter is pointing to that structure and name the structure as well, right? So this is a structure that you need to know, right? So D is going to be, so transmits nerve impulses to the brain. That is going to be your optic nerve, and D is going to be pointing at that. What becomes more or less convex to focus light on the retina, that's going to be a lens, and that is represented by C. What is going to control the amount of light passing through the pupil? That is going to be your iris, and that is labeled by E. What is going to contract and relax to alter the shape of your lens? That is going to be B, your ciliary body containing your ciliary muscles, right? What is going to protect and keep the shape of the eye? That is going to be your sclera, which is pointed to by F. And what converts light energy into nerve impulses? That is going to be your retina, right? Pointed to by G. Next, you have figure 2 showing two eyes labeled A and B under different light conditions, right? So your pupil is one size in bright light and it's going to be another size in dim light. So here they're asking which of the eyes in figure 2, A or B, shows the appearance of the eye exposed to dim light. When you are in dim light, your pupil is going to widen so that more light can enter the eye for a more crisp image to be generated on your retina. So as a result, in dim light, your pupil is going to be wider. Remember, your pupil is the space left in the middle of your iris, right? So your iris is either going to contract or relax in order to control the size of your pupil. So A is going to be showing you how your eye would be set up in dim light. In B is if you're in bright light. If you're in bright light, those light sensitive cells, your rods and your cones that make up your retina, they can become damaged, right? If too much light is coming into the eye. So when you are in bright light, your iris is going to ensure that your pupil is much smaller to reduce the amount of light coming into the eye so that you do not damage your light sensitive cells. You don't want to damage your light sensitive cells that allow you to convert your light stimulus into electrical impulses. So that is a protective function in the eye. So why is this important? It protects or it prevents damage to the structures of your retina. Now this question here, this question is an HSB question, right? But it's a good past paper question that shows you the different settings of your eye or accommodate. I gave away the answer to the question there, but it's showing you the different settings that would your eye set for focusing on distant or near objects. So figure 1 and figure 2 shows light from an object passing through the cornea and lens of an eye, right? So you're seeing here that this lens is thin and this lens is thick, right? The solid lines is showing you light from a distant object. The dotted lines are showing you light from a nearby object. So light from distant objects comes to the eye and they're almost parallel, which means that they're equidistant. Light from nearby objects are diverging when they come to the eye. So because they are coming in a different arrangement, they have to be bent to a different extent, right? Light from distant objects, because they're parallel, they don't need to be bent as much so that they can focus on the retina. You need them to focus on the retina for a clear image to be formed. So when the eye has a thin lens, that facilitates minimal bending of the light to focus it on the retina. However, you also have light coming in from nearby objects, but because the lens is too thin, the light rays are not going to be bent as much. The image forms behind the retina. So you don't see the nearby object is going to look quite blurry while the distant object looks clear. And you could test that out very easily by focusing on something across the room from you and holding your pen in front of your face. If you focus on the object in the distance, the pen, you're going to see the pen, but it's going to look blurry. But then when you focus on the pen, if you try to see the object in the distance, that is going to look blurry. So again, once your eye is set up to, I don't want to use the word, is set up to focus on a nearby object, your distant objects will appear blurry. And when it's set up to focus on a distant object, your nearby objects are going to appear blurry, right? So they said, in which of the two figures, figure 1 or figure 2, is the object being clearly seen? The object here is the one that is nearby, right? So we are focusing on the one with the dotted lines. So you want to select the one where the dotted lines are being focused on the retina. In this case, it's being focused behind the retina. So this image would be blurry, right? So that is going to be figure 2 is the one where where your thicker lens, because the light rays from your nearby object is diverging, you need to, how to tell if it's focusing far or close. You're going to see your parallel light rays being bent too much. This image is forming in front of the retina. So this is going to be blurry. So you want to show that the object, which is the one they want you to focus on, you want the light rays from those to be hitting the retina, which is being shown in figure 2. Right? So this is showing you accommodation in action, right? You do need to be able to identify what muscles need to be relaxed or contracted in order to focus on nearby or distant objects. So I said, describe the physical changes that occur in the eye during the process identified in B part 2. So what are the physical changes that occur in for accommodation to be able to occur? So just 1 second. Throat killing me here. So in order for this to happen, your ciliary muscles must be contracted. When your ciliary muscles are contracted, it reduces the tension on your suspensory ligaments. So your suspensory ligaments, they are relaxed. As a result, your lens is going to be thicker. If your lens is like this, if there's low tension, it's going to want to maintain this more thicker shape. When there is more tension on the suspensory ligaments, it's going to pull on the lens and make it thin, right? So that's how your changes are occurring. So I have this diagram here that makes it a little bit easier for you to see. When you want the lens to be thin or less convex, you need your suspensory ligaments to be contracted. For your suspensory ligaments to be contracted, your ciliary muscles must be relaxed. So in case for when you need your lens to be thin and you're focusing on a distant object, you need to know that your ciliary muscles must be relaxed, your suspensory ligaments must have tension on it, and that will produce a thin lens. If you're focusing on a nearby object, which means you need to bend the light rays a lot more for it to focus on the retina, you're going to need your ciliary muscles to be contracted, which is going to relax your suspensory ligaments and allow your lens to remain more convex to bend the light more, right? So these are, you need to know the settings for your, and when they're asking you, they just said the physical changes in the eye. You have to know that you have to talk about your ciliary muscles, your suspensory ligaments, and your lens. So that in knowing how to structure your answer is things that you need to know unique to this topic, right? Moving on to reproduction. So figure 2 is a diagram of the human female reproductive system, and they want you to label the organs in figure 2 to show where the female gametes or ova are made. So oogenesis, or the production of the female gamete, is going to occur within the ovaries, right? So you're going to have oogenesis occurring there. So oogenesis is a form of gametogenesis. So gametogenesis is how your gametes are being produced. But when you're talking about the female gamete, it's oogenesis. When you're talking about the male gamete, that's going to be spermatogenesis, right? Where would fertilization occur? Fertilization must occur in the fallopian tube or the oviduct. So once the gamete is formed during ovulation, that gamete is going to be released into the fallopian tube, and fertilization must occur here, which means that semen must be deposited in the vagina. It has to swim through the cervix, up the uterus, into the fallopian tube, and meet your ovum in the fallopian tube for fertilization to be able to occur. 3, where implantation of the fertilized gamete would normally occur, that would occur in the endometrium of the uterus, which is the lining of the uterus. Yeah? Miss, repeat that, please. Repeat which one? So, again, the ovary is where, do you have 2 ovaries? That's where your female gamete is going to be produced. Your ovum, during ovulation, 1 month, an ovum will be released here. The next month, this ovary will produce one. So your ovaries take turns every month producing your ovum, right? So during ovulation, it's going to be released into the fallopian tube, and that is going to be where fertilization must occur, right? So I think what you want me to recap, gametogenesis is your production of gametes. When it's the female gamete or the ovum, that is called oogenesis. When it's the male gamete or your sperm, that is called spermatogenesis. Right? So showing your wind pollinated flower, you have to label the different sections. I'm not going to focus on that. All I'm going to highlight here is that when a flower is wind pollinated, your stigma is going to be feathery like this in order to catch pollen from the wind, and the anthers dangle out of the leaf to catch wind so that your pollen could be taken away, right? So you want to focus on that. When it is you have your flower looking like this, it's more likely to be insect or bird or animal pollinated, because both your anther and stigma are going to be within the flower. You're going to see these nice large petals that are going to, and it's going to produce nectar in nectaries to the base of the flower to attract these insects, right? So you need to be able to differentiate between flowers adapted for insect pollination versus wind pollination, right? You need to be able to identify the region of the female part of the plant. So you need to label your stigma, style, ovary, and ovule. So you identify your stigma, your style, your ovary, and your ovule. Your ovule, once fertilized, will develop into your seed. The ovary will develop into the fruit, with the wall of the ovary forming the pericarp of that fruit, right? So the stigma is where your pollen has to land. It will grow a pollen tube, grow down the style, and then enter the ovule at the micropylar end to fertilize the egg cell within the ovule. So that is what happens when pollen lands on the stigma. Once that occurs, this will develop into your seed and the ovary will develop into your fruit. The menstrual cycle, right? Figure 4 illustrates the events that take place during the human menstrual cycle. So this is the uterine cycle, what is happening in the uterus. This is what is happening in your ovaries, right? So that is me giving away part of the answer, because hormone X and Y are your ovarian hormones. And these are influenced by hormones released from your pituitary gland, which would be your luteinizing hormone and your follicle stimulating hormone. Somebody's asking, with the plant, doesn't it need 2 gametes? So the pollen grain actually contains what we call a generative nucleus that produces 2 male gametes. One male gamete is going to fuse with the egg cell, and the other male gamete is going to fuse with this diploid nucleus in the middle. So this produces your endosperm, this is going to produce your embryo, and that is what is going to allow the plant to be able to generate, right? So skipping back here now. So within your ovary, they want you to identify the hormones X and Y and the event occurring at Z. X is going to be your estrogen, right? Estrogen. Take your time with this question. All right. So X is the estrogen, Y is going to be progesterone. So what happens, right? Your pituitary gland is going to release follicle stimulating hormone, and that stimulates follicles within the ovary to start to develop in order to allow the an ovum to be released, right? So once that follicle starts to develop, this follicle produces estrogen. So that's going to cause the level of estrogen to rise, right? So as the follicle develops, it's going to produce estrogen until that follicle develops into a mature follicle, right? The mature follicle, high levels of estrogen triggers or influences your pituitary gland to release large amounts of luteinizing hormone. There's going to be a spike in luteinizing hormone, and that triggers the release of the ovum from this mature follicle. The rest of the follicle remains within the ovary and develops into the corpus luteum, whereas this ovum is released into the fallopian tube, right? So they want to know what the role of estrogen does. So Z is going to be the point where the ovum is being released from the follicle. That is going to be ovulation, and that tends to happen around the mid of the cycle. Now, officially or technically, it occurs 2 weeks before the end of the cycle, but for a woman who has a 28-day cycle, it's going to happen around day 14, right? So this mature follicle is known as your Graafian follicle. I see people knowing terms in the chat. Yes. So that is going to occur around day 14, and luteinizing hormone released from the pituitary gland is what triggers this ovulatory event, right? Within the uterus, the endometrium, or the uterus lining, at the beginning of the cycle, it sheds. So that's when the woman is menstruating and she experiences a period. That lining sheds down to thin, but then once your ovary has started to develop the follicle, it's preparing for this ovum to be released. And if this ovum is fertilized, you want somewhere accommodating for it to implant, right? So the estrogen that is released by a developing follicle tells your endometrium, tells your lining, you need to beef back up, you need to thicken back up, so that if this ovum that we're working on is fertilized, it's going to have somewhere nice and accommodating to implant, right? So that is what causes the lining to build back up. Once ovulation occurs and the rest of this follicle develops into your corpus luteum or your yellow body, right, that produces high levels of progesterone and estrogen. So that is what causes this spike in progesterone. This will persist if the ovum is fertilized. But if this ovum is not fertilized, this is going to disintegrate and stop, and levels of progesterone will drop, and that triggers your lining to shed again. So that menstrual cycle happens over and over and over. It's like a drill for pregnancy, and then if it doesn't happen, it's like false alarm, reset, and it will work like a preparing again for pregnancy, false alarm, it resets, right? So this question just had hormones X and Y, right? But there is also a luteinizing hormone and a follicle stimulating hormone that are released from the pituitary gland, right? So the role of estrogen, it's going to initiate the thickening of the uterine lining. The role of progesterone, it maintains that lining. And the organ that produces the two of them, both are going to be produced by the ovary. However, X is going to be produced by developing follicles, and Y is produced by the corpus luteum. Miss, when does FSH and LH peak? So both FSH and LH peaks just before ovulation. But so your follicle stimulating hormones stimulates the follicles to develop within the ovary. That's its main job, right? Luteinizing hormone, its main job is triggering this ovulation event. And estrogen helps to prepare your lining to be accommodating, and progesterone maintains the thickness of the lining. Miss, for the role, can you say that progesterone inhibits the luteinizing hormone and FSH, and estrogen also inhibits FSH? So you're talking about negative feedback, and that is true. So you can go into detail here, but because it's simply 1-mark questions, you don't have to give such a detailed response, right? How do you know which hormone? So you have to see what is lining up where. Asking if I'm touching on all the topics, I'm touching on the topics that were requested, right? Why is the ovulation? It's showing you the ovum being released from the follicle in the ovary, right? So I'm going to have to move on. Discuss the fate of the following structures if fertilization occurs. If fertilization occurs, the corpus luteum is going to persist and is going to continue producing progesterone and estrogen. The lining is also going to remain thick in order to be accommodating, so that when it arrives, it can implant, right? So moving on to section C, because we do need to get to this topic, which I know is going to take quite a bit, right? So somebody asking, what time is this live finishing? And I would love to say we finish whenever we finish, but I cannot stay as long. Incognito, you missed the I went. So we're going to get into genetics, inheritance, etc. Right? So you have to be able to define certain terms: gene, allele, homozygous, dominant, recessive, that jargon. You do need, yes, I will be touching on natural selection. You do need to know how to define these terms. So a gene is literally a stretch of DNA that is going to code for a particular characteristic or polypeptide. It's the basic unit of inheritance. And an allele are different forms of the gene, right? So different forms of the gene that is for that particular characteristic, right? So it's an alternate form of the same gene. This is not a question, but I need to cover my bases in order to be able to explain. DNA is the genetic material that you're going to find in your nucleus. It can exist in different forms depending on what the cell is doing. Once the cell is an interphase, it's not dividing, it's going about its normal everyday business. It is going to be in the chromatin structure, right? So it looks like thread, noodles, however you want to think of it. It's going to be in the chromatin structure. Only when the cell starts dividing is it going to go into the condensed chromosome structure and adopt this X-shaped arrangement that you're more familiar with. So most of the time, your chromosomes are going to be in the chromatin structure, loose like this, right? Which is loose. But then once the cell starts dividing, either during mitosis or meiosis, then it's going to look like this. Why is in the chat? You get chromatin, you get chromosome. So your chromosome structure, this is going to be the arms of the chromosome. It has a constriction or a narrowing called the centromere, right? Then within the chromatin structure here, you have your homologous pairs. So to introduce the idea of homologous pairs, remember, in order to create you, half of your chromosomes would come from mommy and half of your chromosomes would come from daddy. You get one copy of every chromosome from mommy, you get one copy of every chromosome from daddy. So when fertilization occurs, when the ovum is fertilized by the sperm and the two sets of chromosomes combine, you will have two of every type of chromosome, right? So the copy from mommy and daddy, that is a homologous pair. Yes? Once you have this homologous pair, remember, it's going to have the same genes on it, but it can have different alleles. So for example, you have gene A in this position, gene A in this position, but you can have the big A allele on this homologous chromosome, and you can have the small a allele on this homologous chromosome. Right? So this is your homologous pair here. Before the cell starts to divide, whether it's going to be mitosis or meiosis, it undergoes a process called replication. Replication occurs in interphase before the cell starts to divide, right? So it goes from this single structure, and in replication, an exact copy of both chromosomes are made, but it remains attached at the centromere, right? So you have one copy here, and then you have an exact copy attached at the centromere. We count the number of chromosomes by the number of centromeres, which means that this is 2 chromosomes, this is still 2 chromosomes because we only have 2 centromeres. That's your basics. So let's move into mitosis, right? So you have to know the four stages of mitosis, which is going to be prophase, metaphase, anaphase, and telophase. And so you can remember that by doing PMAT, right? P for prophase, M for metaphase, A for anaphase, T for telophase. Interphase is not a stage in mitosis, being very clear. So I gave you these in your notes, but I'm going to bring the diagram for ease of explanation. So in those boxes are what you expect to happen in each of those phases. But for explaining the phases, I'm just going to use the diagram here for us to be able to move on, right? Interphase, the cell is not dividing. So mitosis begins, it starts here, right? Replication occurs just before division begins. So, in prophase, things that you expect to see happening. Your chromosomes are going to go from the chromatin structure into the chromosome structure. Again, in the chat, interphase is not a stage in mitosis. It's the stage that the cell is in between mitotic divisions, right? It's not that it isn't really part, it's not part, right? So in prophase, what do you expect to happen? Your chromosome shorten and thicken, so it goes from chromatin to your chromosome structure, right? Your nucleolus is going to disintegrate. Your nuclear envelope is going to start to break down, and your centrosomes are going to move to opposite poles of the cell. Your spindle fibers start to form. Your spindle fibers control the movement of your chromosomes the way the strings control your marionette puppets. It's going to be the same thing. It coordinates the dance, right? Then In metaphase, your chromosomes line up on the equator of the cell. 1 second. Right? Your chromosomes line up on the equator of the cell. So that is going to be a distinctive feature of metaphase. In anaphase, the centromeres split. So the first copy, the chromatid, is going to go to one pole, and the other copy is going to go to the other pole. Now that the centromeres have split, we consider them to be separate chromosomes. So when they arrive at the opposite poles, you're going to have telophase occurring, where your nuclear envelope is going to reform around the two groups of chromosomes, and then your cytoplasm is going to be split into two daughter cells. Mitosis is like a photocopying machine. It produces daughter cells that are identical that you started off with. So PMAS is showing you what happens to the dance of your chromosomes and the reforming of the nuclear membrane. Cytokinesis is just the sharing up of the rest of the cell. Right? So moving on, animal cloning is an important mitotic process for living organisms. Explain 3 other ways in which mitosis is important. You need to know why mitosis is important: for growth, for repair, and as a form of reproduction in some organisms. Right? No, I wouldn't be doing any graphs tonight, guys. But I did do a graph in an enzyme question in a previous live, so you could look for that on the Cohen Springer channel, right? Then moving on to meiosis, you have to be able to talk about the stages as well. So again, I'm going to refer, this is all there in the notes for you, but I'm going to refer to, if I could get my diagram. Right, my diagram here to talk about the different stages. Now remember, before meiosis begins, you must have replication occurring, that you're going to have a copy of all of your chromosomes. Meiosis is different from mitosis in that it has 2 divisions. So you go through PMAT times 2. You're going to have prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Then you're going to have prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis again. So this is your cell in interphase. You can see that interphase is where replication is going to occur, and this is before meiosis begins. The diagrams, are you required to know how to draw these diagrams? Yes, they can ask you to draw cells in different stages of mitosis and meiosis, right? Meiosis is happening now, mitosis just went, right? So again, the prophase, metaphase, anaphase, telophase, you're going to expect certain things to happen in each of these stages, and they're very similar to prophase, metaphase, anaphase, and telophase from mitosis. However, meiosis is going to have some unique characteristics, and the differences I'm going to highlight. So you have meiosis 1 and you have meiosis 2. In prophase 1, unlike mitosis, your homologous chromosomes find each other in meiosis, right? This is CSEC biology, right? So in prophase 1, your homologous chromosomes find each other. When they do that, they can undergo a process known as crossing over. So you have your homologous chromosome, your copy from mommy and your copy from daddy. Chromatids can overlap and form a chiasma, which will cause a break and switch. So you end up with one chromosome that is mainly mommy with a little piece of daddy, and a chromosome that is mainly daddy with a little piece of mommy. So that helps to reshuffle alleles between the 2 chromosomes, and it adds to variation in the offspring, the gametes and resulting offspring that is produced. So crossing over is one reason why your daughter cells from meiosis are varied, whereas the daughter cells in mitosis would be identical, right? That happens only in prophase 1. So your nuclear envelope disintegrating, your spindle fibers forming, same yada yada from mitosis. In metaphase 1, your homologous pairs line up on the equator of the cell. So as you can tell, everything in meiosis 1 is done in homologous pairs, right? In anaphase, your anaphase 1, your homologous chromosomes are going to separate. So one homolog is going to go to one pole and the other homolog is going to go to the other pole. Once they arrive, nuclear envelope reforms around each of them. The rest of the cell is going to divide via cytokinesis, and you end up with 2 daughter cells. These 2 daughter cells are haploid. You started off with a diploid cell, and at the end of meiosis 1, you end up with 2 haploid cells. So meiosis 1 is where your number of chromosomes are going to be halved, right? And that is going to be important because if every time gametes fused, it kept doubling the number of chromosomes, it would not be compatible with life, right? So, you know what happens? Like if you have an extra chromosome 21, you end up with Down syndrome, and there are developmental complications with that. So you could tell that having a whole extra set of chromosomes is going to be problematic. Diploid is when you have 2 sets of chromosomes or 2 copies of every chromosome. Haploid is when you have 1 set of chromosomes, right? So the haploid number is going to be, so if the diploid number is 46, your haploid number is going to be half of that, which will be 23, right? These 2 haploid daughter cells then go into meiosis 2. So you're going to have the nuclear envelope disintegrating again. And then you're going to, I think, Miss, slow down. But I have to end the live soon. So I'm trying to fit as much as I can in the live, guys. Right? The nuclear envelope disintegrates. Now you no longer have homologous pairs because the homologous pairs are now in separate, they're now in separate cells, right? So you're going to have now, right? So your individual chromosomes will line up in metaphase 2. You're going to have centromere splitting in anaphase 2, and your chromatids going to opposite poles, right? With this, you end up producing 4 haploid cells now because you had centromere splitting, and now the number of chromosomes, we consider them to be separate chromosomes, right? So what is unique in prophase 1? Your homologous chromosomes find each other. In prophase 2, that can't happen because they're in separate cells. In metaphase 1, homologous pairs line up on the equator of the cell. In metaphase 2, individual chromosomes line up on the equator of the cell. In anaphase 1, homologous pairs separate. In anaphase 2, sister chromatids separate. So you do need to know the difference between what is happening in meiosis 1 versus meiosis 2 and how both of those are different from mitosis, right? One part of the body where meiosis occurs, meiosis is the form of cell division that produces your gametes. All other cells divide by mitosis. Only the cells that produce gametes divide by meiosis. So in the body, where are gametes going to be made in the body? It's going to be made in the ovaries and the testes, right? So either one of those would be correct. And then name the structure within the nucleus that contains your genetic material. That is going to be your chromosome. 2 reasons why meiosis is important. Specifically, meiosis 1 is going to be important because of crossing over that occurs in meiosis 1. Crossing over and random assortment are 2 processes that generate variation in meiosis, so that your 4 daughter cells produced are going to be different from each other compared to your parent cell. That does not happen in mitosis, and that's why you're going to have identical daughter cells from mitosis. The other reason why meiosis is important, that variation increases survivability of the species. It's the only process by which gametes are going to be formed. So you need your gametes for sexual reproduction, and it halves the number of chromosomes, and that happens in meiosis 1 of meiosis, right? Seen some good contributions in the chat, right? Meiosis also occurs in flowering plants. Name the 2 gametes that are going to be produced. So you still have your pollen grains that contain your male gamete. You have 2 male sperm cells in your pollen, and your ovule contains your egg cell, right? Male reproductive system. I wanted to get a diagram for that, but again, just looking up a label diagram would be easy for you to review, right? So here, you do need to be able to compare mitosis and meiosis. What are the differences between the two? In mitosis, no variation is produced. In meiosis, produces variation. Mitosis, 2 daughter cells. Meiosis, 4 daughter cells. Mitosis, diploid cells give you diploid daughter cells. Haploid cells give you haploid daughter cells. Whereas meiosis, you get haploid cells from a diploid parent cell. Guys, if I slow down, I'm not going to be able to touch on inheritance or variation, right? Mitosis produces identical daughter cells. Meiosis, your daughter cells are not identical. Mitosis is cell division for your somatic or all of your body cells. Meiosis produces your gametes or your sex cells, right? So I'm not even going to bother to write out these. I left the annotations, the answers up on how to work this question. What I will refer to you is this link here. I did a separate live with Mr. Springer where we went through step by step your inheritance questions. So you want to go there to this link if you want to go through more questions at a slower pace, right? Because I'm letting you know it's going to go pretty quickly, right? So, 2. A man with normal pigmentation and an albino woman produces 2 children, one of whom is albino. Using appropriate symbols, draw a genetics diagram to show the genotypes of the parents and the expected phenotypes of the offspring. Note that.
I think you might start back to do the up-down thing.
All right, let me switch.
Yeah. So, Miss Akili was supposed to really go 2 minutes ago, so everything right now is just Miss Akili being extra with you. So, there's a lot of research in biology, one of the longer subjects for CXC. But well, Akili, it's up to you. So far as how much you want to go over.
So I tried my best here. Because I feel like I'm teaching a whole crash course again. But I'm gonna go through one basic inheritance question, one sex inheritance question. We're gonna touch on variation, and I feel like that's where we will wrap. Right? So they said, note that albinism is a trait caused by inheritance of a recessive allele. To understand this question, you have to learn the jargon. You have to know what dominant is, what recessive is, what homozygous, heterozygous, what all of those mean, right? So in this case, when they say use appropriate symbols, we get to pick. So I chose A. When they tell you that you could choose the appropriate symbols, you must tell the examiner what those symbols mean. So you have your statements here. You're saying let big A code for the allele for normal pigmentation. That is one phenotype. And I'm saying let small a code for the allele for albinism. And the reason why we're letting small a code for albinism is because they told us it's due to the inheritance of the recessive allele. Recessive traits are only expressed in the absence of a dominant allele. So the only time to be albino is if you are homozygous for the recessive allele, which means that you must have 2 recessive alleles, right? So if it is to be albino, you have to have 2 recessive alleles. It means that the albino woman, we know her genotype, what alleles for this gene that she has 2 small a. And the children, the one who is albino, must also be small a, right? The man with normal pigmentation, he could either be big A or big A small a, right? Both of these genotypes produces an individual with normal pigmentation. However, if it is that this normal man is going to have a child with albinism, he must have a copy of that small a allele to give. So between the two, he cannot be homozygous dominant, because that would mean that all of his children would be normal. It would be impossible for him to have an albino child because the big A allele is dominant. So the only way that a normal man can have an albino child is that he must be heterozygous at that locus, which means that he must have two different alleles, one dominant and one recessive, right? When they say draw a genetics diagram, jump straight into a Punnett square. A Punnett square is going to simply be a 3x3 table, and you write your statements. The parental phenotype, one is normal, one is albino. The genotypes, we already decided that this parent, this man, must be heterozygous, big A, small a. Mommy, we know she's small a, because there's only one way to be albino, right? What gamete types can each parent bring? Now remember, in meiosis, it halves the number of chromosomes, so only one allele from your genotype ends up in a particular gamete. So half of daddy's gamete will have big A alleles, and half will have small a alleles. Mommy only has one type of allele, so all her gametes are going to contain a small a in it. So to work out the possible fertilization combinations, so this gamete could fuse, this type of gamete could fuse with this one, or it could fuse with that one. This one could fuse with this one, or it can fuse with that one. So to work out that in a nice coordinated manner, we're going to do this Punnett square. So we put the possible gamete types that daddy could produce over here. We're going to produce the type of gametes that mommy could produce over here. And following the colored arrows, we work out the possible combination. So if these two come together, it will be heterozygote. If this one and this one comes together, also heterozygote. But when this and this come together here, you get your albino possibility, and here you get your albino possibility. So the possible genotypes that could be produced from these two parents is 2 out of the 4 times they are going to produce big A, small a individuals, and 2 out of the 4 times the individual could be small a, small a. So we work out that genotypic ratio, or that odd, right? So you have a 2 big A, small a to 2 small a genotype, which works out to a 1-to-1 ratio, because we always express ratios in its simplest format, right? So the phenotype associated with big A, small a is going to be normal pigmentation. The phenotype associated with small a is going to be the albino phenotype, and that gives you a 1-to-1 ratio, right? So we have shown the expected phenotypes of the offspring. Again, to go through this at a slower pace, check this link here. I did multiple questions, right? In terms of working out the percentage or the probability, 2 out of the 4 times you could be normal, so that would be 50% chance of being normal, or a 50% chance of being albino, right? The notes are on the Discord. If you use big A for the man, you would have been wrong, right? Because it would have been impossible for a man who is big A to have a child who is albino. Question skipping by quick. Miss, how come you didn't have the father as homozygous? Because if the father was homozygous, all of his gametes would have been big A. And anytime big A meets small a, all of your heterozygotes would have normal pigment. But they had a child who is albino, so we know that he must have had a recessive allele to give. Right? Another example is where with hemophilia. So when the affected gene is located on your X chromosome, which is one of your sex chromosomes, we describe the condition as being sex-linked, right? So with being sex-linked, it means that your different alleles are going to be found on the X chromosome. What makes this complicated is that males cannot be homozygous or heterozygous, because to be male is to have your X chromosome and your Y chromosome. To be female is to have two X chromosomes. So if this gene is on the X chromosome, females will have two copies of this gene, but males can only have one. So females could be homozygous or heterozygous, but males could only be hemizygous because they only have one copy of that X chromosome. So if they get a bad copy or the allele associated with disease, they will have the condition. If they get the good copy, they're good. If they get the bad copy, they're bad, right? So they told us that this disease, hemophilia, is caused by an X-linked recessive gene. So X-linked means that it's on the X chromosome, and recessive means that you have to have two recessive alleles, or basically the dominant allele must be absent in order to express the condition, right? So they said 2 parents with normal phenotype have a hemophiliac child. So your parents' phenotype would be normal and normal. There's only one way for daddy to be normal. Again, he cannot carry a recessive allele. So for daddy to be normal, he must have the normal allele, right? So we worked out the different codings here for you in blue. So daddy's genotype is X big HY. Big H for normal clotting. Hemophilia is the lack or the inability of the blood to clot, right? Or abnormal clotting. So daddy has this normal allele on his X. Mommy is also normal, but if they're going to have a hemophiliac child, that recessive allele for hemophilia has to be somewhere, right? Mommy can be a carrier because mommy has 2 X's. So one of her X's would be normal and the other would be recessive. There are 2 ways for mommy to be normal: 2 dominant or a dominant and a recessive. But because she's going to have a hemophiliac child, she must have that hemophiliac allele to give, right? So mommy must be a heterozygote. She must have a normal allele, which allows her to be normal, but she must have a recessive allele to be able to give to her offspring. So the gamete types that daddy could produce, half his gametes are going to have the X with the normal allele, and half his gametes would have Y. Mommy, half her gametes will have the normal allele, and half her gametes are going to have the recessive allele that causes hemophilia. So we put the two types of gametes from mommy on one part of the Punnett square, the 2 parts from daddy on the other side of the Punnett square, and we put the different combinations, right? This is going to produce a normal female, has 2 normal copies of X. This is going to produce a carrier female. She would be normal because she has a normal X, but this recessive allele means that she's carrying the allele for this condition, but she's not expressing it. This will be a normal male, right? 1 X, and that X is normal, the male is going to be normal. But in this case, this male would get that recessive allele from mommy, and because he only has 1 allele, he does not have a backup. So therefore, he is going to express the condition, and he is going to be a hemophiliac, right? So in this case, it's only possible for a male child to be a hemophiliac if the mother is a carrier, right? Again, if you want to go through this slowly, go back to this link on this page of your notes, right? Or just search Cohen Springer channel for, I think it's CSEC Biology revision from 2023, right? Hemophilia is a disorder caused by a defective allele, whereas persons aren't able, their blood isn't able to clot properly, right? Mr. Springer will have to say when the live is available. I'm not too sure.
The live will be available immediately. As soon as you finish, it's available. It's still follow the link on this group.
Miss, how come some people, their first child is be a boy instead of a girl where the X is always leading? Not too sure where. So The Y chromosome is responsible for maleness, right? So once a Y chromosome is present, it means that the child is going to develop into a male. Miss, can you explain the biological reason for homologous chromosome Y not copying and carrying the allele? Your Y chromosome is much shorter than the X chromosome, so it just has much less genes on it, right? This question was an old question, but it covered, so all the time we were talking about alleles that exhibited complete dominance, which means that one allele completely masked the other. Your dominant allele, your big A, masked the small a, so you still had the phenotype of the big A. But you can have other types of in relationships between the alleles. You can have incomplete dominance and you can have co-dominance. And this question highlighted both. So it said, complete the chart in figure 3 using letters and words as appropriate to show the inheritance of flower color between two pure breeding varieties of impatiens when they are crossed. The heterozygote phenotype is pink. So two true breeding means, or pure breeding, true breeding means that you have two of the same alleles, right? They must be homozygous for that allele, right? We have 46 chromosomes for the person who add in the chat. So to be red, they must have two R alleles. To be white, you must have two W alleles. But the relationship between these alleles is that one, it does not completely mask the other. So that's why they're using capital letters for both, right? They told us that the heterozygote, which means when you have two alleles coming together, one R and one W, the phenotype is going to be pink. So the phenotype of your heterozygote is different from either parent. It's a blend of both of your parents, right? So this phenomenon is known as incomplete dominance, where a blend of red and white will give you pink. So your gametes here would be 2R from this parent, 2W from this parent. When one gamete from each come together, because there's only one combination that would result, all of the F1 would be heterozygotes, have an R and a W, right? F1 will, the F1 phenotype, the phenotype of the heterozygote, the characteristic that will be expressed based on the genotype, that's what your phenotype is going to be pink. So the question goes on to say, well, if you then cross these heterozygotes, which means that you do pink by pink, or you do RW by RW, which is, we can say that it was selfed, it was crossed with another of the same genotype. All of those mean a way of saying the same thing. They ask, what would be the phenotypic ratio of flower color in the offspring? So you put your gamete types here, and these are your four possibilities here that you will get. So you will get one big R, two big R, big W, and two big W, big W. This would be red, this would be pink, and this would be white, right? So you write down that ratio in the chat, 1 red to 2 pink to 1 white. When expressing genotypic ratios or phenotypic ratios, always write the phenotype or genotype associated with the number in the ratio. It minimizes error. What type of genetic variation is shown by flower color here? This is going to be incomplete dominance, where one allele does not completely mask the other, but there's a blend of the two, right? So this characteristic, flower color, is controlled by one gene that has two alleles, and these two alleles exhibiting incomplete dominance produces 3 phenotypes. This produces discontinuous variation. Discontinuous variation is when your phenotypes fall into set classes. So a common example, so in this case, it's either the flower is red, or it's pink, or it's white. Those are the 3 categories, and so that is discontinuous variation. Another example of discontinuous variation in humans would be a blood type. It's either your blood type A, blood type B, blood type AB, or blood type O, right? There is no blend, etc. you fall into one of those 4 categories. That's going to be a discontinuous variation. Continuous variation would mean that there are multiple possibilities for the phenotype. There are subtle variations in the phenotype. So, for example, height, weight, etc. Those are going to be continuous variation, right? So then it goes on to say the alleles of the ABO blood group in humans show codominance. Codominance is when both alleles contribute to the phenotype of the heterozygote, where the phenotype of the heterozygote, you're going to see the phenotype associated with each individual allele, right? So how does this codominance differ from the incomplete dominance shown by the impatiens flower, right? Your blood type is determined by one gene with 3 alleles. You have your A allele, B allele, and your O allele. The A allele and the B allele are codominant. One is not dominant to the other. One is not stronger than the other. Both A and B, however, are dominant to O. So A is dominant to O, B is dominant to O, but A and B are codominant, right? So to be type A, you could either have 2 A alleles or an A allele and an O allele, where the A allele will be dominant to the O allele and still result in you being type A. To be type B, 2 Bs or a B and an O. To be type O, which is the recessive allele, there's only one way to be type O, which is to be OO. To be type AB, you have to have an A allele and a B allele. So there's only one type to be AB, right? So codominance, both phenotypes associated with each allele is seen in a heterozygote, right? So if flower color, you have incomplete dominance, which is a blend or pink, which on this slide, I think I left it in your notes. Where is it? Right. So with the pink, the IN in pink, remember when there's a blend, when it's pink, it's incomplete dominance, which is going to be different from codominance. Codominance, when the phenotypes associated with each allele is shown, it means that if the flower color in this flower was showing codominance, the flowers would have been red and white. So codominance is red and white. Incomplete dominance is a blend, which would be pink, right? So they ask you in part 2, if it was showing codominance, what color the heterozygote would be? It would be red and white, right? So these are different types of relationships between the alleles that govern a different gene loci, right? So guys, I had to go through this quickly. After this, go and watch the next live where I taught this inheritance topic by itself at a much slower pace. If you don't get it, check that one and catch up, right? This, I was showing you how inheritance occurs in blood type. Again, that is covered in that live, so you can check that there. This question from 2023 was asking, why do siblings, even though they come from the same parents, why do they show differences? Why that the A is not on the syllabus, guys, right? If they ask you why siblings differ, it's because of meiosis. It's because meiosis produces variation due to crossing over, etc. So your gametes produced by your parents happen as a result of meiosis. Variation is going to be introduced. Crossing over and random assortment explains that. And then they will have various genotypes at each of the loci that allows them to be different. This question is in the notes. I'm going to skip past, which would be the similarities and differences between mutation and sexual reproduction. We went through continuous and discontinuous variation. This is one I want to get to. An ecologist identified 2 species with identical niches, which means that they occupy the same role in the ecosystem. Define species. As species, you have to hit certain criteria. You must say similar characteristics. You must say that the species, they must be able to interbreed. And when they interbreed, the offspring must be fertile. You must hit those 3 things in the you must hit those 3 things in your definition of species. Suggest 2 possible outcomes if the species were to compete for the same resources. And you can give one. You can have extinction of the weaker species. You can have resource partitioning where they decide to share the resources. Or one species just relocates, right? 2 ways in which speciation can occur. It either occurs with some physical barrier, or which is called geographical isolation or allopatric speciation, or it happens within the same geographic area, but differences in behavior, which will be your sympatric speciation. That's your major difference between the 2. So one involves a physical barrier, the other doesn't. So you have to be able to differentiate between those 2. Natural selection. When you hear natural selection, I want you to think survival of the fittest, right? 4 minutes, 11, I have to go at 11. You have survival of the fittest, which means that the organisms that have the best alleles, they have characteristics that give them an advantage to survive. They are going to live, they are going to reproduce, and they're going to pass on those alleles to the next generation. So natural selection, think survival of the fittest, but you must have passing on of the alleles to the next generation, right? So that is going to be to outline the theory, Darwin's theory of natural selection. That is it in an outline, right? Natural selection assists in speciation, outline 3 advantages and 2 disadvantages of natural selection. So rather than focusing on this question, let's think about natural selection, right? I will give you an example. If I had a mutation, and mutations are the way in which new alleles are generated, I had a mutation that allows me to be able to breathe underwater, right? All of us here, all of us above water, and we good. So my mutation of being able to breathe underwater does not give me an advantage to survive, right? Because the environment doesn't require me to be able to breathe underwater. Now, knock on wood, some tsunami come covering the whole Caribbean, all of we underwater. If you don't have that mutation that allows you to breathe underwater, you're not going to survive. So the environmental conditions change, whereas the entire surface of the earth is covered in water, but my mutation of being able to breathe underwater gives me an advantage in this new environmental condition, which means that I live. Now, if I live, I could then have children and pass on that allele of being able to breathe to the next generation. That is natural selection in action, right? So that happening over and over and over from generation to generation can eventually lead to new species being generated, right? So you can outline your advantages and disadvantages. Natural selection is about the having characteristics best suited for survival as being the criteria about who gets to pass on their allele to the next generation or not. Artificial selection is when man decides which organism is going to pass on its alleles based on characteristics important to man, not necessarily important to the survival of the species. That is the major difference between artificial selection and natural selection. But again, I have that explained for you guys here, but time is against me. It is literally 10:59. So the last thing I want to touch on is genetic engineering. And one use of genetic engineering, that this is not even the question, this one, right? One use of genetic engineering is being able to mass produce insulin. Diabetics, persons who either don't produce enough insulin or their body does not respond to the insulin that they have produced, they need to take in extra insulin, right? Getting insulin was very difficult because it used to rely on the meat trade. So when they slaughtered pigs and cattle, they would use the insulin from those organisms for humans. It was in short supply, it depended on the meat trade, and it was very inconsistent in supply, which would have ramped up the price of it. So we have been able to exploit E. coli to produce human insulin so that we can give it to humans, right? So how does this work? Within your human pancreas cell, within the nucleus of that cell, you're going to have DNA. DNA, the stretch of DNA or the gene for human insulin, we're going to want to cut that out. And we would use something called a restriction enzyme to cut it out. Then the E. coli bacteria naturally has small circular rings of DNA called plasmids. We are going to take that plasmid out of the bacterial cell. We cut that plasmid with the same restriction enzyme, so the ends of the human insulin gene and the ends of the plasmid match. So we insert the human gene for insulin production into the plasmid, and we form what we call recombinant DNA. This recombinant plasmid, you then insert back into the bacterial cell in a process known as transformation. And now, as that bacterial cell is expressing its own genes, it's also going to express the human gene for insulin. So it means that once we culture or ferment the bacterial cells and increase the number of them containing the human gene for insulin, it means that we're going to get a whole bunch of bacterial cells that are producing human insulin. We can then package that insulin and sell it for diabetics to be able to use. Right? So that is 1101, guys. Again, the questions that I didn't do in detail, you have a copy of that in the notes. You can get that note on the Discord, the CSEC Biology Discord. You can download it from there. It's freely available to you guys. I am at the point where my voice is like struggling. But
That that insulin thing, that's why your PhD on.
no, my PhD is on genetics of diabetes, but not on exploiting bacteria to produce the insulin. By trying to figure out why our genes increase our likelihood of having diabetes.
Oh, okay. All right, Audio, send up some love for Miss Day for coming through. That was 1 hour, and then the next hour and a half. So some good revision there. I and all learn. Well, revisited some of my biology topics there. I don't know how I'm gonna do with all this knowledge now. So thanks, Miss. Somali, I've seen some people who was here in maths and every single live, only doing every subject that we have, boy. Then I've seen some names from the student hub, from Miss class, from Miss Crystal class. So we wish you all the best in biology exams. We pray that, excuse me, whatever you all study will come.
Mm-hmm.
No weird exams, that you'll get a nice solid exams that test how much you know. And then there would be cooking tomorrow and not be the cooked. All right. So without any further talk, Akili, you have anything to add?
No, all the best, make the most of the time that you have.
Um, it was you who passed, and I was in Cuba today. I think it's your car seat drive past, and that was you.
It was my car, but it was my husband. It was John.
Oh,
He's
okay,
sorry.
okay. Yeah. So all right, Alia, bless up. So
Bye.
Saturday we coming back with Cam. We may see if we can squeeze in some maths probability tomorrow. And physics. I know how the physics happening. Maybe Sunday night we'll hit some physics. Love and blessings, everybody. Enjoy your exam tomorrow. I will hear about Ali in the exam watch on TikTok later.
Bye.